And we're live.
Great. Well, welcome back, everybody, to Elasmo Week. I'm your host for the week, Dr. David Schiffman. Elasmo Week is an outreach event about the science of elasmobranchs, sharks, skates, and rays, and the scientists who study them. And this week is all about diversity. The goal is to highlight the diversity of scientists, the many ways of doing elasmobranch research, and to show off as many weird and wonderful species as possible every year. Elasmo Week wants to make a platform where real scientists can share their love and work with the public. It's my pleasure to introduce tonight Dr. Sora Kim, who is an assistant professor in life and environmental sciences at the University of California, Merced. She explores the interactions between environmental change and biotic response in modern and ancient ecosystems. The strength of her work is its interdisciplinarity, which spans paleontology, physiology, ecology, geochemistry, and paleoclimate. Some current projects include, why did Megalodon go extinct? How did the Drake Passage, opening 50 million years ago, impact shark ecology and global climate? How does ecological niche differ among deep-sea sharks? What environmental and ecological factors affect body size distributions? She also has non-shark projects that include the impact of climate change on historical and ancient wolf ecology, and exploration of food web assembly and disassembly, and method development to enhance stable isotope applications. A fundamental tenet in her research program is to promote diversity and, more importantly, engage in the deep work needed for equity and inclusion. Take it away, Sora.
Hi, thank you. First, before I get started with my talk, I really want to acknowledge and stand in solidarity with Scholar Strike, which is happening today. I didn't feel that I could shirk on my responsibilities to Elasmo Week, but Scholar Strike is an effort for academics to sort of stand with Colin Kaepernick and the NBA, the WNBA, and other sports figures who have, decided to take a stand against police brutality and racial injustice, and especially anti-Black racism. And so we should acknowledge that, and especially in relation to STEM and academia, and that these are still very pervasive white spaces and can oftentimes be unwelcoming to minorities of race, gender, and sexual orientation. And so we, in celebrating the diversity of sharks and its researchers, should be part of those that push back against these white and oppressive institutions. So with that, I will get started with sharks. So my work is starting to sort of traverse more towards to paleo sharks, although I started off doing a lot more with modern sharks. And so the title of my talk today is Deciphering Climate, Oceanography, and Ecology from Fossil Shark Teeth. And here are some images. There's images of folks looking for fossils in Antarctica, some members of my lab doing some lab work and weighing out samples, modern Carcharias taurus, sand tiger shark, and a fossil tooth from about 50 million years ago collected in Antarctica. Let's see here. Uh-oh, okay. So this sort of story collection is going to be talking about the protagonist, who is sharks, and they represent a successful evolutionary strategy with a 400+ million year fossil record and survival through multiple mass extinction events. And so sharks are highlighted in teal here, and you can see that they evolved in the late Silurian is when we first see fossils, and really the only bottleneck and period of time where they were hammered in terms of their diversity is at the Permian-Triassic boundary. But otherwise, they were able to get through a lot of mass extinctions, unlike other organisms. So we have to ask ourselves, what can we actually learn from this fossil record and the longevity of this taxonomical group? And here I have an image by Ray Troll, excellent artist and also aficionado of sharks, paleontology especially, and different depictions of extant and extinct sharks. And so the period of time that I'm going to be talking about is the Eocene. The Eocene, as shown here in the orange box, it captures a transition from greenhouse to icehouse climates, but actually remains quite enigmatic. So here in this figure is the climate record for the last approximately 65 million years. And this is all from oxygen isotope analysis from phorians, which are these tiny little protist shells that we find in deep sea sediments. And if you analyze these calcium carbonate tests, then for the calcium carbonate, the oxygen within them tells us something about temperature. And here is an estimate of ice-free temperature along a secondary y-axis. And you can see that during the Eocene, within this box, that we have really warm temperatures, some of the warmest, and that as time moves towards the present, that we actually get this gradual and steady cooling throughout time as ice sheets form both in Antarctica and also in the Northern Hemisphere. And why do we actually care about this topic? And so for a lot of paleontologists, we talk about how the key is the, okay, sorry. I am trying to get us, get this gray box out of the way. We talk about how the key is the past. The past is the key to understanding the present. And so today, many of you are probably aware that modern shark populations are actually declining quite rapidly. And here in a study in 2010 by Francesco Ferretti, along the x-axis you have year, and on the y-axis you have catch per unit effort. And you can see that there are these severe declines happening from the shark fishing nets in Durban, South Africa, for most of these large charismatic shark taxa. And so in particular, I'm interested in looking at Stratolamia macrota, and the reason why is because of where these fossil shark teeth are found. So Stratolamia macrota is very similar to its modern analog, Carcharias taurus, and they have this preference, it seems, for temperate water with a large spatial distribution. And so on the top here with the blue box, you can see the modern Carcharias taurus teeth and the distribution highlighted in blue. And then this map from Nebo et al. Uses satellite tagging analysis data. And you can see that they have this seasonal migration from the bays of Massachusetts all the way down to Florida. And so in the fossil record, we have this image from Padilla et al. These are teeth found in the Eocene Arctic on Banks Island. And those teeth are very similar in shape to the modern Carcharias taurus teeth. The map here, all the dots are different fossil localities for the extinct Stratolamia macrota and where they've been found. So you can see that they really are widely distributed all the way from the Arctic to the Antarctic, much larger distribution than even the modern sand tiger. And when you go into fossil collections, you actually find these boxes with hundreds of teeth, sometimes thousands. And so for the Eocene Arctic, there are approximately 18,000 shark teeth that have been collected from Banks Island. And of those 18,000 teeth, only 4 are not Stratolamia macrota. So that's telling you something about how dominant this taxa is within the Eocene. And so how can these fossil shark teeth contribute to our understanding of Eocene paleoclimate and paleoceanography is the core of my research questions. So I'm going to give you 3 stories today. One is evidence for a closed Eocene Arctic Ocean. Second is going to be navigation of the early Drake Passage. And 3 is going to be the impacts of temperature and migration on size distribution. So the Eocene Arctic was an ice-free ecosystem, and the terrestrial components of this ecosystem, they included temperate flora and fauna. This is an artist rendition from Eberly et al. In 2011, and you can see Jalen looking quite cold in the Arctic. She's a professor at CU Boulder. She's the one that introduced me to Stratolemia macrota in the Eocene. And so in this artist rendition, you can see sort of this lush temperate forest where you have a coryphodon. It's kind of a hippo-like mammal, and then you have these small mammals in the understory, but also primates in the trees. And so that is very different from what the modern day Arctic looks like, which is where Jalen is huddling there. And for the marine record, Lomonosov Ridge is actually the only marine record that we have for the Eocene Arctic. And based on various geochemical proxies, we have low, warm salinity conditions. And so what the reconstruction looks like by Ron Blakely here 50 million years ago is that the Eocene Arctic Ocean may have been completely enclosed and been sort of like a Lake Arctica has been a nickname given to the area. And you can see a star there where Lomonosov Ridge is reconstructed to be. And the reason why it's so hard to get records, marine records today, although this could be changing with climate change, is that the Arctic Ocean is full of ice and it takes a long time to drill these cores. And so this is the middle image here is a drilling rig that I think that it went out in 2005, and they had to have 3 icebreakers constantly circling the drilling rig in order for it to not get stuck and frozen into the ice. And so with the oceanic core that they were able to get, they found low salinity tolerant dinocysts, which is shown in the blue line there. And then in the purple line, they were able to get back Tex 86, which is a temperature indicator. And so you look at these temperatures in the purple line, and you see that this scale bar on the x-axis here is ranging from 15 to 25 degrees Celsius. So that's pretty warm and very different from the modern Arctic Ocean, even with its warming that's happening present day. But it's a pretty splotchy record. We don't necessarily have a continuous record from this period. And so what I use is called oxygen isotopes. And oxygen isotope composition, it is it's preserved in the hard parts of organisms, and it reflects environmental water temperature and the environmental water's delta O18 value. So these are a series of 2 equations, and what we analyze here is the delta O18 value of calcium carbonate, or in hydroxyapatite, there's carbonate and phosphate, also in enamel or enameloid in the case of sharks, but it is codependent on both temperature and the delta O18 value of the water. So what does that mean? And so what it means is that if you were to take the offset, so the difference between the delta O18 value of the carbonate or the phosphate minus water, that spacing is actually proportional to the temperature. And so this is a cartoon sort of diagram showing what that relationship looks like. But it really is sort of this confounding factor between the delta O18 value of the water and also temperature. And so what actually controls the delta O18 value of the environmental water is 2 things. One is Rayleigh distillation and the other is salinity. So Rayleigh distillation is the process by which oxygen isotopes or deuterium isotopes will differentiate between the heavy and the light hydrogen isotopes, hydrogen and deuterium. And so with wind patterns, that moisture originates near the equator. And then as you go to more northern latitudes, there's Hadley cell circulation that's happening over the ocean that transports water vapor north. And as that, every process of distillation actually means that you get less and less of the oxygen 18, because as water is evaporating, oxygen 16 is selected for, and that is what is getting transported to these high latitudes. So by the time you get to somewhere like the Arctic, in the ice or snow, your delta O18 values are close to -20 to -40 per mil. And what this means in terms of salinity is that these fresh water, all the water that's deposited on land, it also has low delta 18 values. And so as you have mixing between saline water, where salinity would be close to 35, and seawater has a delta 18 value approximately of 0 per mil. So this is where seawater would lie. Meteoric water, or snow and rain that has fallen from the sky, it would have a salinity of 0. It would be fresh water, and it has relatively low delta 18 values. So as you have mixing between this meteoric water with a salinity of 0 and seawater with a salinity of 35, your delta 18 values also co-vary along this gradient. And this has been documented, actually. And so salinity and delta 18 values co-vary, and there's actually a global pattern across their relationship. So here on the x-axis is salinity. This is an old graph, so it has per mil there. And then on the y-axis is the delta 18 value relative to SMO, which is standard mean ocean water, the standard. And so these are different transects that have been done in different oceans. So in the Eastern Equatorial Pacific, the Gulf of Mexico, the Sargasso Sea. And so they just measured the salinity and the delta 18 value of water offshore. And they found this covariation. And what this means is that when salinity is 0, so the y-intercept, that would be the delta 18 value of the fresh water coming into the system. And so the second graph is on the x-axis, it is the slope of these individual lines. I've highlighted 2 in gold and green here. And so on the x-axis is the slope of these lines, and along the y-axis is the delta 18 of that freshwater diluent, so the intercept of the lines that are in the left graph. And so this gives us a model that can give us some predictions and correspond delta 18 values and salinity. And so to first establish the approach, we tested it with modern sand tigers. Folks have analyzed the delta 18 values in fossil shark teeth, but it's really not necessarily to get at salinity, and it's pretty limited in terms of the ecological interpretation and approach to it. And so for this project, we got samples from modern sand tigers from Dr. Dwayne Fox at Delaware State University. He has a team out there that collects modern sand tigers and tags them. And then we did stable isotope analysis in the lab. And here are some students who are visiting my lab from William Patterson University in New Jersey for a couple weeks during the summer, prepping samples, and we run them. And then the third component of this project was actually to put them into a Bayesian model. And so this model was able to put in things like sea surface temperature and the variation in sea surface temperature, and then also the delta 18 values of the seawater as it's been documented. And the modeling was done by Dr. Dave Bell, who's now with the USDA. And so this is just a conceptual diagram of what the model looked like. And we looked at the analysis of modern sand tiger shark teeth to look at the method's accuracy. And so the histogram here represents the predicted salinity for the modern sand tiger sharks in Delaware Bay. So it's about 30 to 40. And modern seawater variation is shown as the triangle and the error associated with it. So we sort of predict higher salinity than we thought, but it turns out that even though these sharks were caught in Delaware Bay, that some of their teeth may have formed somewhere else along their migratory pathway. And if that happened to capture something that was happening off of Florida or in the Bahamas area, that there is actually really high salinity in those waters. And so then when we add in the data from the Eocene Arctic, the salinity estimates that we have, they actually indicate brackish conditions and that there's a very high freshwater input. And so here the modern data are shown in blue again. And then the samples from Lomonosov Ridge, those were actually taken from fish bones. That is in white here. And then the sharks are here in the gold. So these sand tigers from the Arctic Ocean about 55 million years ago or so, they're showing salinities on average of about 12.5 or 13, which is really, really low and somewhat disturbing that the Arctic Ocean was that enclosed and had that high of freshwater inputs, and sort of rings true to something that we might think about today in terms of warming and the melting of the permafrost and sea ice. So brackish Eocene Arctic Ocean, it supports this idea of a closed or narrowed oceanic gateway, and it really has implications for thermohaline circulation patterns. And so this was a study done by Roberts et al. In 2009. And in this study, they were trying to test whether the Turgay Strait and the Greenland-Norway Seaway, how open versus closed that they were. And the outputs of these models in terms of the isotopes, they match almost precisely with this scenario of a closed Greenland-Norway seaway and a very narrowed Turgay Strait. So that was something that we did not necessarily have insight into before. So next, in our journey of how can shark teeth contribute to our understanding of Eocene paleoclimate and paleoceanography, we're going to look at the navigation of the early Drake Passage. So this is one of my ongoing projects right now. I don't know if you guys think very much about this time period. It's a really long time ago, but Antarctica is really central to a lot of explanations for the greenhouse-icehouse transition that I was telling you about earlier. And part of that is because today, Antarctica is thermally isolated. So here in this right-hand figure, you can see that there's this Antarctic circumpolar current that goes around Antarctica. And so it is really thermally isolated from the warmer waters north of there. And that happened because the Drake Passage, which is shown in this 4 panels here from 50 million years to 30 million years ago, that the tip of South America and the Antarctic Peninsula used to be connected, or at least sort of like this archipelago formation. And as the tectonic plates moved apart and created the Drake Passage and it deepened and widened, that water was able to pass through there. And so what do we see from there? The paleoecology and paleoceanography are actually preserved in the fossil record. We actually have, you can see here in the panel A, that we have the Drake Passage. This arrow is pointing to the tip of the Antarctic Peninsula, and near that tip is Seymour Island. And Seymour Island has these different groupings called telms. They're called the tertiary Eocene Meseta units, but everybody calls them just telms. And within these telms, we're able to find really amazing fossils. You can see some bivalve shells. These from Ruggiero et al. Are penguin bones. And then here you can also see a sea star. So even invertebrates are very well preserved, which is amazing. And the sand tiger shark teeth are very abundant. And so I think that they include ecological and environmental information that we can harness. And so we've already talked a little bit about delta 18 values being related to temperature. Here, salinity isn't as big of an issue because we're actually in a fully marine environment. Some other tools that I used here were to look at Neodymium isotopes, which track ocean circulation, and then to look at tooth size as a proxy for body size. And then by measuring multiple teeth for their size to get size distributions of the population that was here. So to talk a little bit more about the methods, Neodymium, it actually uptakes during early diagenesis. And so that's when, after the shark tooth is lost in sort of the first maybe hundreds or 1,000 years, that it captures the Neodymium isotope value of the pore water in the sediment sort of surface layer. And so for fossil fish teeth, it's thought to be sort of imprinted and then not lost afterwards. And the reason why neodymium isotopes allows us to look at ocean circulation patterns is because neodymium isotopes reflect the crustal weathering rates of an oceanic basin. So different oceanic basins have different types of rocks within them, and there are different ages to these rocks. And so because the oceanic basins have different ages, they actually have different neodymium isotope values. So this is work done with Howie Scher at the University of South Carolina. And some of his work in 2015, this is from the Tasman Gateway. So that's actually on the other side of Antarctica, where we also need it to be open in order for this Antarctic circumpolar current to form, that he was able to find this really sort of sharp drop off at about 30 million years ago, indicating that there had been a major change in restructuring in the ocean current system. So the next method that I mentioned was to look at body size. And I'm interested in body size because it reflects the energy balance and it integrates sort of a physiological, environmental, and ontogenetic factors. And so body size is a way to get a handle on the available resources, the temperature, and the physiology of the animals. And so Tina Feynman, who worked on this on her master's project at CU Boulder, she found that if you look at the she measured the maximum height of anterior teeth in sand tiger sharks, and based on work that was done by Kenshu Shimada, found that the crown height is related to total length. So this is Dr. Shimada's work that relates the crown height of the tooth to the total length of the animal. And each individual position in a shark, so here's a diagram from Capetta of different tooth positions. So each individual tooth position has one of these regressions associated with it. And contrary to previous studies, the temperature data from the oxygen isotopes of Stridolaemia macrota, it shows a relatively stable temperature throughout the La Meseta formation. And so here in this graph on the right side is a sea surface temperature that is estimated by TEX 86, it's a geochemical proxy, as well as clumped isotope analysis from shells. And you can see that sort of there's this cooling as you go from 44 to about 40 million years ago. And that's also tracked in TEX 86. But if you look at the data from sharks, which is this more vertical panel right here, in each telm or time period, this is older here in telm 2, this is more recent in telm 5, you can see that there is really no pattern. There's a lot of variation, but no actual real pattern that's happening. And then if we look at the neodymium isotope values, we actually see that there is this, that at no point do we actually have a completely Indian/Atlantic Ocean end member value, that even in telm 2, that we already have some inputs of the Pacific Ocean, it looks like. And here I have a picture of my student Molly Carnes. We went to Sweden together to visit Dr. Thomas Morse at the Swedish Museum of Natural History, where we looked at a lot of shark teeth. And so what we're getting out of this data is that although temperature was constant, there's actually already inputs from the Pacific Basin as early as telm 2. So it's not that the Drake Passage is completely closed actually during this time. And when we look at the diversity of the shark teeth, S. macrota, it really dominates the shark record in the La Meseta Formation. And so we were able to look at hundreds of teeth, just anterior teeth from the La Meseta Formation. And this is with Dr. Jürgen Kriwet at the University of Vienna. We measured hundreds of teeth, and we found that body size is actually quite stable. So here along the x-axis, we have the log of the anterior tooth crown height, which is a proxy for body size. And then on the y-axis, we have the density. So these are probability density functions. And for the specimens from the La Meseta Formation in Antarctica, they are the red, yellow, green, and blue as we've had them. But I also show the size distribution of the modern sand tigers from Delaware Bay. You can see that the distributions are actually quite similar, but these ancient sharks actually have a bit wider of a distribution. But essentially the mean is almost the same between the modern and the paleo and throughout this entire time period of the paleo record. So how did body size and temperature remain constant despite an early opening of the Drake Passage, as we have suggested by Epsilon Neodymium? So this was kind of a big question that we had. And so to pursue this, we came up with a couple of hypotheses. So our first hypothesis was that maybe Stridolaemia macrota migrated across the continental shelf from the Antarctic Peninsula to southern Chile or somewhere in southern South America during an early Drake Passage. And so if we have this proto-Drake Passage where it's breaking apart, but it's still like fairly shallow continental shelf, it's totally possible that Stridolaemia macrota could have actually migrated through this area. And the support for this is that extant sand tigers, that they seasonally migrate. So I showed you this graph before from satellite tracking data from Dwayne Fox's tagging program. And they found that, that even though their sharks were caught in Delaware Bay, they go all the way down to Florida, and they mostly stay in relatively shallow and warm waters along the continental shelf. Some of the larger females, they go out to the edge of the continental shelf, but not very many of them. So our second hypothesis was actually based on another study that was published by Zhu et al. In 2020. And so they did this climate model, this global paleoclimate model, and they actually modeled what the temperature and the oxygen isotope value of the water would be if CO2 concentrations were 3 times and 6 times pre-industrial levels. And so that is what is shown in panels A and B and D and E. And then we used those, if you remember from like many slides ago, those equations that I showed you, that the delta 18 value from calcium carbonate or from phosphate in enamel and enameloid, that it is dependent on temperature, but also the delta 18 value of the water. So we sort of did a reverse design of those equations to predict what the delta 18 values of phosphate would be if they had formed in these conditions. And so our second hypothesis was that the Eocene Southern Ocean was warm due to high CO2 concentrations. So even though the Drake Passage was open and that there may have been sort of a proto-Antarctic circumpolar current, that things were kept warm because of these elevated CO2 concentrations. And in support of that, from these climate simulations, the predictions that we were able to get for the delta 18 value of phosphate is actually almost exactly with what we see from our fossil shark data. So here in the blues, purple, and orange are the delta 18 phosphate values that you've seen before across different tomes. And in this panel B is actually the seasonal variation in what the predicted delta 18 phosphate values would be across months. And so you can see that the variation that we have for 6 times the partial pressure CO2 and then 3 times the partial pressure CO2, that envelope sort of captures the full range of delta 18 phosphate values that we actually see in our fossil shark teeth. So from the second part, we think that temperature preference is likely a conserved trait, as we can see in the modern sharks at Delaware Bay, and then also these fossil sand tigers from Antarctica. And the number of shark teeth in the fossil record allows us to reconstruct body size distributions. And it remains constant for millions of years in the Eocene Antarctic. And then finally, we think that these shark teeth really offer us the best vantage of the Drake Passage opening. These are the fossil specimens and the data that are the closest to where the Drake Passage was actually opening, and they are suggesting a very warm Southern Ocean during this time period. So I'm going to do a quick time check. I'm doing pretty good. And so last but not least, to look at how can shark teeth contribute to our understanding of Eocene paleoclimate and paleoceanography. Now we're going to go straight to just looking at the impacts of temperature and migration on size distribution. And so there's this idea that that there are differences in body size related to latitudinal changes or temperature, and so we're curious if that holds true for sharks. And the experimental design here was to look at shark teeth from both high and mid latitude sites and nearshore versus open marine sites. And so we have 4 different fossil localities, and it's sort of a block design, classic block design, that we have mid latitude in this first row, and then high latitude in the second row. And we have nearshore shown in green, shades of green, and then this open marine shown in shades of blue. And you can see this distribution across here. And the other red dots are other fossil localities. And then in terms of time, this is how they're distributed along time. And all of this data was collected by Sarah Zeichner, who was an undergrad at Caltech, or was an undergrad at University of Chicago, but is now a grad student at Caltech, and Dina Feynman, who was doing a master's at CU Boulder, but now is in Victoria University, and she's actually a geomorphologist. So doing something totally different and not shark related at all. And so when from these different fossil localities, as shown in the same block design here, but I've gone ahead and named the actual geological formations, that we looked at a lot of different teeth. And so the smallest sample size that we have is 292 teeth from Texas in the Stone City Formation. But you can see that these sample size numbers are huge. And so we are able to get very robust mean and standard deviations from these collections. And what we see is that body size distribution patterns, that they seem to vary with latitude and the extent of the marine influence. And so in green, we have the sites that are much more terrestrial influence, and that is determined by the co-occurring fossils on the site. And then in the darker blue are the marine sites, or the 2 shades of blue are the marine sites. And so they seem to have larger shark teeth. And anecdotally, this is what sort of paleontologists have said for a while. The other thing that you might notice is that these high latitude sites, which are the dark green and the dark blue, so this dark green is here, and then the dark blue here, that their distributions are also quite different. So maybe even not all high latitude sites are equal. And so to look at this, we were just, I was kind of curious of like, what does temperature and migration have to play into this? And so this was work done with Justin Yankel, who is now at UC Merced, but I went to grad school at UC Santa Cruz. And so he's a theoretical ecologist and does a lot of modeling. And so I asked him if he'd be interested in collaborating with me on this. And so he developed this model. And our question was, what do shark teeth reflect about population level processes related to temperature and migration? So modern ecologists oftentimes think about, the number of sharks, but we translated sharks into essentially teeth to get at the fossil record. Our model has 4 different components to it. There is a birth component, death component, a growth component, and this growth is dependent on the temperature, and then a migration component, so longer and shorter migrations. So birth and death, the rates are actually fixed, but growth rate is temperature dependent. So for birth, we had about 4.5 female pups per female per year post maturation. And for the death rate, we had 0.18 per year. And the growth would depend on metabolic rate. And so this is an approximate model that we found, that the mass at birth was about 8 kilograms, the asymptotic mass, is about 110 kilograms, and that we have differences for warm versus cool water. And so we've played with these numbers, and it turns out that they're not particularly sensitive to the situation. And so with migration, we can look at the distance and the timing to adjust for these paleo conditions based on what we think might have been happening. And so we have like sort of a high latitude migration, then a mid latitude migration. In terms of the migration window, we think about that the migration to the nursery might happen in sort of this distribution of time. And then the migration to the adult site, that might be that not the whole population moves all at once, right? That the juveniles, maybe some individuals take longer than others in terms of what size that they would want to move over to the adult site. And so this is what some of the outputs look like, that we have time in years along the x-axis and then the population size along the y-axis. In red, we have the nursery site. In blue, we have the adult site. And then in green, we have sort of the combined signal of the adults and the juveniles. And so populations among the sites, they cycle across annual migrations. And then we sort of look at the teeth that are lost to simulate the fossil record. And so for tooth loss, we look at 2 teeth per 40 days. And that is to think about mortality-induced accumulation. And so the model output patterns, they actually vary with latitude and life stage, and we're still trying to like figure all of this out because it's pretty complicated. But we see that the temperature really affects the distribution of the larger sharks. So in these figures, we have, I'm showing you a mid-latitude site. So in terms of temperature, that they're pretty temperate. Warm, and then the migration would be about 1,500 kilometers. And so in the mid-latitude site, that this is this top series for the nursery site, the adult site, and the combined. And then at the lower 3 panels is at high latitude. So same migration distance, but actually colder temperatures. And so when you have colder temperatures, the nursery site, it actually doesn't change in its size distribution, but it gets like this little interesting little hump here. And then for the adult site, these sharks are actually quite a bit smaller. And so you look at the frequency of these larger shark teeth in the mid-latitude site, and there's a lot more of them. And then again here, when you look at the combined between the nursery and the adult site, you get this sort of bimodal distribution almost between, which is representing the juveniles and the adults. And so migration seems to have actually the largest influence on body size distribution in terms of mean and modes. And so in each here in these figures, that on the left side, we have the juvenile sites. On the right side, we have adult sites. And the top row is the mean tooth size. So here you can see that at the juvenile site, the largest teeth that we have, no matter what the migration window is for the juveniles or the adults, that the largest teeth that we have at the juvenile site is about 15 millimeters. And that is somewhat predetermined by our model, saying that juveniles move to the adult site at a certain size range, maturity point. And then at the adult site, though, you can see that that we have the largest teeth here, where we have a small juvenile migration window. So we limit how many juveniles come to the adult site, but the adult migration window, like when the adults can come and can leave to the nursery, it doesn't matter as much as how we get the largest shark teeth. And then we also looked at that the distance between the modes. And so there's that like sort of like little hump that happened at the bimodal distribution. And so if you look at that, we looked at it at the juvenile site and at the adult site. And then along the x-axis here is the juvenile migration window, and on the y-axis is the adult migration window. So you can see that the frequency of, or the distance between these two modes, which is what this color scale is here, that the two modes are far apart in this area, in the juvenile site, but then in the adult site, you get these two modes a lot more frequently, but they're usually fairly close together, except when the adult migration window is small, then you actually get these two modes separating from one another. And so the question that we were curious about is, can we use a simple population dynamic model to help us understand habitat use, body size distribution, and nursery usage? And so now we're in the process of trying to match sort of what would be the most likely, match likelihoods to get at what would be the parameterization of the migration windows at the adult and juvenile site to sort of match what these outputs are of our empirical teeth distributions. So to bring it all together, to talk about lessons learned from Eocene Sand Tiger sharks in terms of paleoceanography and paleoclimate, looks like that the Eocene Arctic Ocean was a relatively closed system and it had low salinity. That Seymour Island records the earliest Drake Passage opening based on Neodymium isotope values, and that the coastal shelf near Antarctica was warm during the early Eocene despite an open Drake Passage. And then why were sand tigers so successful? And it's mostly likely due to ecological plasticity. And this might be the case for a lot of especially mesopredator sharks. In terms of paleoecology, that sand tigers can live in waters with prolonged reduced salinity, that sand tigers in the Eocene Antarctic potentially migrated across a proto-Drake Passage, and this would ameliorate the effects of climate change. And then demographic models can help us infer the influence of temperature, seasonality, and migration on ancient shark size distributions. So with that, I have a lot of acknowledgements. I covered a lot of time and spatial gradient here. For collections, I had samples from the Canadian Museum of Nature, the Carnegie Museum of Natural History, the Whiteside Natural History Museum, and then also the UC Museum of Paleontology, as well as the Paleontological Research Institution, Swedish Museum of Natural History, and the Argentine Antarctic Institute. There were multiple funding sources. This was a lot of different projects that I presented for you, and they are majority funded by NSF from various programs. And so this is just my last slide in terms of the take-home messages. And so thank you very much. I hope that you feel that that this was a cool shark show in terms of this illustration by Ray Troll from his book Sharkabet, which is an awesome book.
Great, thank you very much, Sora. That was awesome. Everyone who's watching on YouTube, we're going to end this broadcast in just a minute, but just to know that there's another Elasmo Week talk coming up on a different YouTube, or the same YouTube channel, but a different video in just a few moments here, and we'll be doing a live question and answer session with all of the Elasmo Week speakers on Sunday evening. So if you have a question, leave it on the YouTube video that you're watching now, or tweet it at Elasmo Week, or sometimes it takes a minute to get going, Francisco, sorry. is the connecting Zoom to the YouTube, and which usually works, but I can't tell if it's working from here. So Jasmine's checking on her end.